Good day, everyone. Isn't it a wonderful day to learn a new topic in biology? That is why my group and I will be presenting you on a very interesting topic entitled Fungi, the Dikarya Subkingdom. My group consists of four members. The first presenter will be me, Priya. The second presenter will be Amira. The third presenter will be Naufal. And the last presenter will be Balkish. So without further ado, let's begin. As you can see in the pictures here, Ascomycota has different shapes, sizes, and structures. Now let us dive deeper into the world of Ascomycota. Ascomycota phylum is also known as sac fungi, and there are 64,000 species. Their morphological type is divided into two, which is multicellular molds and single-celled yeast. Ascomycetes also play two important roles, one in the ecological role and another in the economic role. In the ecologic role, ascomycetes play a role in recycling, where it digests material that do not easily decompose, and this includes cellulose, lignin, and collagen. Ascomycetes also stimulate beneficial relationships with plants, algae, and some animals. In the economic role, yeast, which is an example of ascomycetes, produce food. Ascomycetes can also cause food spoilage and produce toxin. Ascomycetes also is used in the production of antibiotics where it treats fungal infections. Ascomycetes are also used in the food industries, and there are three major examples, which is the yeast Saccharomyces cerevisiae, genus Aspergillus, and the penicillium. Yeast Saccharomyces cerevisiae, also known as yeast, it is used in baking and brewing industries. Sugar, with the presence of yeast, undergoes fermentation, and sugar and yeast, when it's used in the making of bread, the bread rises because because CO2 gas is released. And also sugar and yeast also undergoes fermentation to produce ethanol, which is a type of alcohol, and this alcohol is used to obtain liquor, wine, and beer. Moving on to penicillium, it is used in the cheese industries to make various kinds of cheese, such as blue cheese, Roquefort, Camembert, Brie, and Stilton. It's also used to make an antibiotic, which we commonly know as Panadol. Lastly, we have genus Aspergillus, which is used to produce soy sauce. It's also used in the Japanese food called miso and is produced by the fermentation of soybeans. Genus Aspergillus is also used to make Japanese beer, which we know as sake. Citric acid and gallic acid, which is an example of genus Aspergillus, is used as additives in foods, inks, medicines, dyes, plastic, toothpaste, soap, and chewing gum. Let's take a look at the disadvantages of Ascomycota in humans. When we are talking about the disadvantages in humans, we are focusing on the diseases that can be caused in humans. One of them is Candida albicans. An unbalanced Candida albicans and other microorganisms can cause oral thrush in newborns and vaginal infections in women. If you're wondering what's oral thrush, you can see it in the picture here. It occurs on the surface of the tongue. Moving on, we have Tinnitus. What is Tinnitus? It is a mold-like fungi that causes infection. And what kind of infections that can it cause? It can cause athlete foot, jog itch, and ringworm. And what are the symptoms? There will be itching, peeling skin, and raised inflammation. Some people are more prone to fungal infection. Who are these people? These people are patients with AIDS and patients with immunocompromised conditions. So what are these patients with immunocompromised condition? These patients have low immune system where their body cannot fight against diseases. These fungi also creates a difficulty in creating medicines. Why? Because there are many similarities between fungal and human cells. So over here, that is this scan that shows a fungal infection that occurs in immunocompromised patients. Now let's look at the advantages of Ascomycota. Yes, there are advantages of them despite the disadvantages. Firstly, Ascomycota have antibiotic and microbial properties. Genus Penicillium, as an example, produces antibiotic penicillin, which is a treatment of bacterial infections. Cyclosporin, on the other hand, controls immune system to prevent organ rejection after a transplant. Next, Ascomycota is used as a model organism. Why? It contains genome information. For example, Aspergillus nulandens. It is a safe model for more dangerous related species. Next, we have Neurospora crassa, which is used to develop the one gene, one enzyme hypothesis. Lastly, we have Saccharomyces, which is a single-celled eukaryote with genes similar to other eukaryotic organisms, and it can be easily manipulated in the lab. That's all from me explaining to you about Ascomycota.
Hello everyone. So I'm going to continue the presentation with the reproduction in Ascomycota and also about the 3D model. So in Ascomycota, there are two types of reproduction, which are the asexual reproduction and sexual reproduction. In asexual reproduction, this is the most common type of reproduction that occur in this type of fungi. Usually, yeast will reproduce by asexual budding, which is the small cell will pinches off until it grows to a full size. Also, mold will produce a spore called conidia or conidia spore. Conidia spore is a spore that produced by sac or club fungi during asexual reproduction. So next, I am going to talk about the sexual reproduction in Ascomycota. So to explain this, I'm going to explain with our group 3D model over here, which is, this is Morchella esculenta. So this Morchella over here has three structure, which are ascocarp, rhizopid, and pid. So as you can see, ascocarp located above the fungi. Rhizopid is this ruffle-like structure, and pid located between the rhizopid. So to get a better view of the structure, you can refer it over here. So the process of sexual reproduction in Ascomycota, firstly, the sac fungi will reproduce sexually by produce asci. So asci is a finger-like sac, and it is protected by a sterile hyphae. In a fruiting body. Fruiting body is a exposed cup, as you can see in this Morchella over here. So in the the fruiting body has characteristics such as it is cup shape, and also it is stacked with pid, like in this Morchella esculenta. So within the ascus, the dikaryotic hyphae will terminate forming the asci, where the nuclei will fuse to create diploid cell that undergo meiosis to form a new ascocarp. Therefore, in the morel is the ascocarp of Morchella. The asci will born on the ridge of pid. In a simpler term, the asci will transported and carried on the ridge of pid.
Next, I'll be talking about Basidiomycota, or known as club fungi. At the background, we can see some example of this fungi. This one is called puffball, and this one is called stinghorn. And we can see at the leaf there are some orange spots, which is called rust fungi, which is actually a disease for plants, but I will get more into it later. A little introduction about this club fungi of phylum Basidiomycota. It consists of around 31,000 species, such as mushrooms, toadstools, puffballs, shelf fungi, stinghorns, and 75 species that are considered poisonous. An example is the death cap mushroom. There are also plant diseases such as smuts and rust. These fungi usually reproduce sexually, but there are some reproduce asexually. This reproduction involves the fusion of two haploid hyphae of the opposite mating types, forming a dikaryotic N plus N mycelium. This dikaryotic mycelium forms fruiting bodies called basidiocarps, or we known as mushrooms. Portabella and shiitake mushrooms are savored as food by humans. This is also an example of the club fungi, and these fungi contain club-shaped structures called basidium, and it performs nuclear fusion followed by meiosis, producing basidiospores. And these basidiospores are windblown and produce new haploid mycelia, and new mushroom will grow at that area. It is not encouraged for amateurs to collect mushroom in the wild because there are certain poisonous species such as red and yellow amanitas and the death angel mushroom, which cause 90% fatalities to mushroom poisoning. This poison is really dangerous. It could cause serious problem. The poison will inhibit the RNA polymerase that could cause liver and kidney damage. It can also cause abdominal pain, vomiting, delirium, and hallucination. This is the red amanitas, which is actually really easy to distinguish because of the bright color. And this one is the death angel mushroom, or Amanita phalloides, which look like a normal mushroom. That's why it's really dangerous. Sometimes people mistake with the normal mushroom. Next, I'll be talking about the life cycle of a mushroom. First of all, the cytoplasmic fusion. The cytoplasmic fusion will result in dikaryotic mycelium. At this stage, we call it the button stage of a mushroom. And this dikaryotic mycelium produces mushrooms, or we call it basidiocarps. We can see the cup-like structures, we call it cap. It is supported by a stalk. And then basidia form on gills of a mushroom. Under the cap, we can see some lines, which is called gills, and basidia will form on there. And then nuclear fusion occurred, resulting in a diploid nucleus. And then meiosis occurs and basidiospores are produced. These basidiospores are released by the wind, and then these basidiospores are germinated, resulting in a haploid hyphae. And there will be two haploid hyphae fusing and continuing the cycle of a mushroom. Next is smuts and rust. Smuts and rust are club fungi that parasitize cereal crops. These have great economic importance because of annual crop losses. The smut and rust do not form fruiting bodies or basidiocarps, and its spores are really small and numerous. Life cycle of rust often requires two different plant host species, and the way to control it is to eradicate the alternate host. And for wheat rust, another way to control it is by breeding new resistant strains of wheat, which requires continuous development because rust can also mutate. This is the example of smuts and rust. The white thing on the picture is the smut, and the next picture we can see a single strand of wheat, and the orange thing is the rust. That is all for my Basidiomycota slides, and this is the references for my slide.
So, to make us easier understand about the life cycle of a mushroom, I will show you a 3D model. So, this is a 3D model. This is the cap. Under the cap have the gills. If you can see, have the lines under the cap. And the orange is annulus and the white is stalk. So, as we know, mycelium will produce a mushroom or basidiocarps. So, basidia will form on gills of a mushroom, then nuclear fusion resulting in diploid nucleus. Meiosis occurs and basidiospores are produced. Then basidiospores are released, then germination of spores result in haploid hyphae. Then cytoplasmic fusion resulting in dikaryotic mycelium. Then we can have other mushroom. So, Thank you for watching our video. Then we hope that you can understand this topic well. Thank you guys. Bye.